Hello, this is Dr. Martin, and we're beginning week 8 of the semester. Week 8 is the halfway point of the semester. This is a 16-week course, so this is halfway. Now, the remaining 8 weeks go really fast after this because one of them is spring break, and we don't do any work on spring break. So the second half of this semester goes really fast. Last week we covered chapter 3, the cell, and all of the parts of the cell, all the organelles of the cell. And this is the next chapter, chapter 8, skipping all the way to chapter 8. We're going to talk about how cells replicate, how cells divide. So let's get right to it. Cells don't just, remember this, cells don't just grow bigger and bigger and bigger and bigger. Instead, they reach a certain point in size and then they divide. Remember, there's a triggering mechanism inside the nucleus that triggers, or there's a threshold that the cell crosses that begins this process. Remember, we talked about some things. The nucleus controls this process. Also, the surface area relationship between the membrane of the cell and the volume of the internal part of the cell. That ratio is very important. And when it gets to get kind of maxed, then the nucleus triggers the cell division process. Remember, we talked a little bit about that last week, and we're going to talk a lot more about that triggering mechanism today. So cells divide. They don't get bigger and bigger and bigger. They simply divide before they get too big. And this is a classic cell right here, nice micrographed picture of a cell. All cells divide. All cells go through cell division. Now, eukaryotic cells like in our body, eukaryotic cells divide by a process we call mitosis. You might remember that term from high school biology. So eukaryotic cells divide by mitosis. Once fertilization occurs, sperm cell, egg cell come together, then all cells begin to go through this mitosis, beginning with one cell, ending up with about 6 to 7 trillion cells as adults. And that's what we call mitosis. Now, we also produce egg cells and sperm cells in the testes of males, the ovaries of females, but that's a very specific process called meiosis. In meiosis, we are producing specifically cells that will eventually be fertilized and eventually develop into a zygote or a person. Hey, plants do this as well. Fungi do this as well. So this is the sexual life cycle, sexual reproduction. So the difference between these two processes, meiosis occurs in the testes of males, ovaries of females, also the ovaries of plants, but this specifically results in sex cells, what we call gametes, sperm cells, egg cells, pollen grains. Mitosis is what occurs in every higher organism, plants, fungus, even animals. So this is two different processes. One is much more specific to sex cells. One is much more general to all cells, or what we call body cells. And remember, in meiosis, this is where two sex cells will fuse, what we call fertilization, sperm cells, egg cells, and join DNA, or, part of the DNA is contributed by the mother, parts contributed by the father, and then that's what we call fertilization. But that's different from mitosis. So let's go to mitosis then. All cells go through mitosis, even the most small, simple protozoans go through mitosis. Notice this one cell on the far left. It's a process where one cell divides into two separate cells. The key here is that the genetic information remains exact. These two cells on the far left, genetically, they are clones of the original cell. We call this original cell the parent cell. We call these two cells at the end of mitosis the daughter cells. And the two daughter cells have the exact genetic information as the parent cell. Now think about this for a second. If they don't, if mistakes occurred, remember what we call that? That's a mutation. And so it's very important that cells guard internally against changes or mistakes in copying the DNA of our cells. But this is mitosis, starting with a parent cell, ending up with two daughter cells. This occurs in body, what we call body cells. That's the cells of our bodies. We also call those somatic cells, or the cells of a plant, the plant body. This is mitosis. It's starting with one and going rapidly and ending up in a trillion cells. Whereas if we produce a lot of cells, it makes sense that we also have to remove some cells. Cells have a life cycle. The cells that began in your mother's uterus, those cells have died and have gone away. So we are constantly going through mitosis, but we are also constantly losing cells. Cell death is what we call apoptosis. It's removing cells or cells that die. It's what we call cell death. And if you're going to produce a trillion cells in a period of a month, you have to remove about a trillion cells in the period of a month, and that's what we call apoptosis. Well, the most important thing in mitosis are the chromosomes, the DNA. All of those other cell parts, mitochondria, endoplasmic reticulum, the Golgi apparatus, the ribosomes, hey, all those will be manufactured from the instructions on DNA. So all of those can be made later. But during mitosis, the key is making copies of every chromosome, and that those chromosomes are copied exactly. We don't like mistakes. Now, just as a side note, there are mistakes. In fact, about 6% of male sperm will have some type of genetic mutation from the parent cell. So yes, it's not an exact process, but it is really close. It's about 95-94% accurate in their copying. So you have 46 chromosomes in all of your body cells. That's the parent cell. When it divides into two cells, we want 46 chromosomes in those two daughter cells, and we want it to be exact. That's what we call our genome. So we really take care of these chromosomes. Now, that's eukaryotic cells. Prokaryotic cells divide by what we call binary fission. Remember, they don't have a nucleus. They're chromosome, singular chromosome. You have 46, bacterial cells, 1. Yours are linear. Bacterial cells are circular. And it's not enclosed in a nucleus. So they still replicate that DNA molecule, but then they go ahead and separate into two, starting with one parent cell, ending up with two daughter cells, and these two daughter cells have an exact replica of that original DNA molecule. But that's what we call binary fission. It's a little different than in higher organisms like eukaryotes. So there's a singular chromosome. It has to be replicated, and then eventually we end up with two cells. The cell volume internally splits into two. It's what we call binary fission. So one parent cell divides into two daughter cells, and this is bacteria and the single chromosome in bacteria. Whereas we in eukaryotes, our cells replicate by mitosis. The cell on the left here is not replicating. Here's the nucleus. Remember, the membrane surrounds the nucleus, but this cell over here, it's getting ready to divide. And we have a name for this appearance of this cell that's getting ready to divide. Every cell in your body has 46 chromosomes, your body cells. We need to replicate or copy all 46 of those chromosomes before we divide the cell. That's the first thing that happens in eukaryotic cells. It looks like a pile, your DNA in your cell looks like a pile of loose yarn in a normal cell not dividing. But in a cell that is dividing, we condense that DNA. We like a slinky, we condense it and twist it upon itself because this has to replicate or double in number, double in content if we're going to divide it. Now think about this. If you have a parent cell, one parent cell, it's a muscle cell, it's going to divide into two cells. It makes sense that if you start with 46 chromosomes, we have to double that material because the two daughter cells are going to have copies of those original 46 chromosomes. So we have to double the material, double the DNA in content, in size, and in number in order to get back to 46 chromosomes. So this is a cell that is in normal cell functioning. This is one that is literally going through cell division. The DNA is condensed. We can actually see it. We can see the chromosomes here under the microscope. And that's what happens when a cell is dividing. Now, every cell has a life cycle. Now, this is what we call the cell cycle. And there's a lot of events that occur in a cell before it's going through dividing. So we divide, as scientists, we divide a cell into phases. It's going to go through. Now, cells don't do this, and in their mind, they're not thinking, okay, I'm going to go from one phase to the next, to the next, to the next. No, we do it as scientists so that we can study cells. And so we classify cells going through various phases of their life. The orange here is what we call interphase. We'll talk about these. In fact, you remember these names. Probably you'll recall these from high school biology. But here's a phase, what we call interphase. It's where a cell is living in its normal life cycle. But here in the blue, this is cell division. This is mitosis. This is where a cell is beginning to divide. And so let's walk through some of these phases of a cell's life cycle. Here's the orange. This is what we call interphase. It's the life cycle of a cell in between divisions. We call it interphase. It's not dividing. It's carrying on its normal function. If it's a muscle cell, it's doing what muscle cells do. If it's a nerve cell, it's doing what nerve cells do. It's what we call interphase. It's the phase in between divisions. And that's the area in orange here. There are three phases of interphase. There's G1, S, and G2. G1 is where the cell is carrying on its normal functions. It's growing, it's maturing. If it's a muscle cell, it's doing what muscle cells do. If it's a skin cell, it's doing what skin cells do. It's going through its normal functions. But what we've talked about is once cells increase and grow in size, that there's something that happens inside that nucleus that triggers a division process. It's ready to, maybe size, it may be the ratio between the cell membrane and the interior volume, but something in the nucleus triggers the division process. And that's what this line is right here. It's a threshold. And the first thing that cells do when they're getting ready to divide is they replicate their chromosomes. They make a copy of their chromosomes. We double the material, we double the content, the volume of chromosomes. We don't care about all of the other cell parts. We'll make those later, but the chromosomes are crucial. So that's what we call the S phase. That's where your cell synthesizes a copy of all 46 chromosomes. All of them are copied exactly. We don't like mistakes. We know intuitively that there's going to be mistakes, but most of them are not harmful. Most of them are harmless. And so, in fact, that's kind of how evolution probably occurs, is through the process of mistakes or changes in cell material. But the first thing your cell does is it makes a copy of every chromosome in the nucleus. G2 is when the rest of the cell begins to ramp up the machinery to divide into two. That's what we call the G2 phase. A lot of material has to happen here. It takes a lot of energy for it to happen. The mitochondria really produce a lot of energy. The ribosomes produce a lot of energy. The Golgi apparatus produces a lot of cholesterol and membrane material. So all of this is what we call interphase. It's the phase of a cell's life in between divisions. So we have the G1 phase, and then we have a G0 phase where it continues to function. But then we have the S phase where we synthesize the entire genome of that cell in the nucleus. And then G2, the rest of the cell begins to ramp up the machinery for the division process. Right here, here's this threshold here where a cell begins to divide. And it goes through the blue sections here. We have names for all of these phases of a cell, but cells aren't doing this. We just do it as scientists because it's easy to identify them. We call this mitosis. This is the division process. And the end of it is where the cell is going to divide or pinch into two, which is what we call cytokinesis. Cyto meaning cell, kinesis meaning tearing apart or splitting. You remember these phases probably from high school. Let's walk through them really quickly. We're not going to spend a lot of time on these, but so here's a cell. It's ready to divide. Here's the nucleus. The chromosomes inside the nucleus have now replicated. So we have double the material. We have eventually what we call a prophase. This is the parts of mitosis. We have a prometaphase, we have a metaphase, we have an anaphase, we have a telophase, and at the end of mitosis, we have two cells, daughter cells, with the chromosomes that have been replicated, or the genome has been replicated, so that these two daughter cells have the exact information genetically as that parent cell did. They are essentially clones. So these processes are pretty easy to follow. Prophase is the very first part. This is where we can actually see on a microscope in the nucleus. We can see the chromosomes. The nucleus is still intact here. This is the cell. Here's the nucleus. Here's a plant cell here, kind of square, kind of like a block. Here's the nucleus. Look, we can see those chromosomes in there. They become condensed and kind of fat and kind of chunky and easy to see. Also, the membrane of the nucleus begins to kind of fade away. Enzymes begin to eat away at this nuclear membrane. Then also we enter what's called a prometaphase, or technically we skip towards a metaphase here. And but this is where all of those copies of the chromosomes, you have 46, this cell just has 4 chromosomes, but this is where the nucleus is gone, the chromosomes line up across the middle of the cell. This is called the middle, or meta. We sometimes call this the equator of the cell. Look at this actual animal cell here. Look at those. It looks like somebody took a Sharpie and drew some black marks across the middle of the cell. This is still just one cell. And here's the chromosomes that are lining up across the middle of the cell. Now, it looks a little different in plant cells because the chromosomes are a little longer and they kind of spread out, but they're lined up across the middle of the cell. Also, we see these fibers. They're protein fibers that extend out from centrioles, Remember, we talked about those in the last chapter, and they attach to these chromosomes. They kind of serve as scaffolding or structure, and these spindle fibers attach to these chromosomes. This is what we call metaphase. Meta means middle. Then we enter what's called anaphase, where the two copies of the chromosomes split apart and begin to move towards opposite ends of the cell. Hey, we're still one cell, but we are dividing up the chromosomes that have doubled or copied. One copy of this chromosome goes towards this end. One copy of this chromosome goes to the other end. Remember, we double the material and then we separate them. And that's how we divide the chromosomes during mitosis. Still just one cell, but look at this animal cell here. One cell, the chromosomes have separated and moved apart. Here it is in a plant cell. Really cool microscope pictures. And we see these pictures in lab. We actually look at these under a microscope. But this is what we call anaphase, where the copied chromosomes pull apart and move towards opposite ends of the cell. Eventually, they move towards way apart. Nuclei membranes begin to form around those separated chromosomes. Here it is here too. Look at this hourglass figure eight appearance. See that? The cell is beginning to pinch in between these two groups of chromosomes that have pulled apart. This is what we call telophase. Nuclei begin to form around those chromosomes. Here and here, the centrioles are at opposite ends. The cell begins to pinch in here to separate. In plant cells, they have a very rigid cell wall here. And so they can't just pinch in the sides. Instead, they form a cell plate across the middle here between these two chromosomes. Look, nuclei are beginning to form again here around these two groups. So what we've done is we've taken 46 chromosomes, doubled them, and then eventually split them apart. And now we're going to split the cell in between them. That's what we call telophase. So this is a cell in telophase. So here it is again. We call this a cleavage here, a cleavage furrow, this hourglass appearance where the cell begins to split in the middle. Here it is, an actual micrograph or a microscope photograph of a cell that is pinching into two. The chromosomes have pulled apart, nuclei are forming around them. Here's the plant cell forming a cell plate in the middle. And that's how a cell goes from one parent cell to two daughter cells. They copy the chromosomes, and then the cell begins to divide. First, there's prophase, where the chromosomes condense and become very fat. The spindle fiber begins to form. The nucleus around the chromosomes disintegrates. Then we have metaphase, where the chromosomes line up across the middle of the cell. Then we have anaphase, where the copied chromosomes pull apart. And then we have telophase, where the cell splits in between those two groups of chromosomes. Nuclei form around them, and we have two daughter cells whose chromosomes are exact copies of that parent cell. That's mitosis. Now remember, mitosis is just the dividing part of the cell's life. All of the time in between mitosis is what we call interphase, the phase in between divisions, G1, S, G2. So make sure you kind of know the general occurrences in each of these phases of a cell's life or the life cycle. What we're going to do next in chapter 8 is we're going to talk a little bit about abnormal cell divisions. Remember what we call that? We call that cancer or abnormal cell growth. We'll do that in the second lecture of chapter 8.